So, hello and welcome to episode 5 of CCLG's Research Talks for Parents and Public. I'm Ellie Wilkinson and my job is to share research with our supporters. So firstly, I want to say thank you for joining us today, and let's start with some housekeeping. So we'll be holding a Q&A session at the end of the talk, so please do submit your questions whenever you think of one using the Q&A button, which should be in the middle of your screen at the bottom. If you'd like to use the chat function, make sure you've set the to drop down to everyone, or people may not see your message. This webinar is being recorded and will be available to watch publicly on our YouTube channel. We will send out the link when it's ready to watch. So this month's talk is called How Has Research Helped Children with Leukemia, and is being led by Dr. Lisa Russell. So Lisa is a senior childhood cancer lecturer and researcher from Newcastle University. She's an expert in the field of leukemia genetics and wants to find a single treatment that would work for a specific group of children with leukemia. Lisa has been working in leukemia research for almost 20 years and has 2 research projects funded through CCLG, one of which is funded by Ruby's Live Kindly Live Loudly Fund, and I can see a few different people from that have joined us today. Hi. So without further ado, I'm going to hand over to Lisa.
Okay, so let me share my screen with you. Okay, so can you see that okay, Ellie? Okay, and I will just add a pointer because I usually like to wave my pointer around. So, okay, so thank you ever so much for the introduction, Ellie, and it's a real privilege to be here this evening to speak to you all about how cancer research has helped children with leukemia. So first of all, let's have a little bit of an introduction and talk about where leukemia develops. So leukemia develops in our bones, in the bone marrow, in the centre of our bones. And so when we look at a healthy bone marrow sample, it should have lots of different cell types in there. So we should have some stem cells. Now, these are really cool cells because they can produce any of the other blood cells that we find in our bone marrow. We can then have red blood cells. So now these are really important for binding the oxygen that we breathe in. And then they deliver that oxygen around our body to all of our organs. We then have a large number of different white blood cells. And so our white blood cells form our defense system. So they form our immune system. And there's lots of different cell types because they need to do lots of different jobs. Some white blood cells can detect that maybe a bacteria or a virus has entered into our body. Some will coat those viruses or bacteria and shout for help from other white blood cells. And other blood cells will provide memory within our immune system. So if we are exposed to that virus again in a couple of years' time, we may not even know that we've been exposed to it because our white blood cells have retained that memory, and they will actually attack that virus before we get any symptoms and know about it. And the last type of cells are platelets. So these cells have the job of clotting our blood. So if we fall over and cut ourselves, whilst we will initially bleed, it will eventually stop. And that's because these platelets will form clots at the site of damage and stop the blood from coming out. So how does this look in a child that has leukemia? Well, they still have all of these different cell types, but they have an imbalance in the number of these particular cells. So we will still see stem cells. We will see fewer red blood cells. The biggest change that we see is in the white blood cells. So rather than having a few of each type of white blood cell, one white blood cell kind of takes over. So this white blood cell will suddenly gain an advantage over the healthier white blood cells, and it will be able to divide really quickly. So we can end up with lots of one type of white blood cell in the bone marrow. Also, these white blood cells get stuck at a specific stage in their development. So when we think of adults, we develop. So we're born as babies, we then become children, teenagers, and then adults. And there's some jobs that we can only do as an adult, say drive a car or get a job. And that's the same for white blood cells. They need to get to their adulthood in order to be able to undertake their proper function. But in the white blood cells of children with leukemia, they get stuck partway there. So not only do we have lots of one type of white blood cell, we have white blood cells that can't actually perform their function. And so now we've got a real imbalance in our immune system. So we've got not enough of lots of different white blood cells to be able to function properly. And we've also got fewer platelets as well. So we can link this with this knowledge, we can link this to some of the symptoms that we observe. So children with leukemia can experience tiredness and fatigue, and that's because they have fewer red blood cells. They're carrying less oxygen around their body. They can also have frequent or severe infections, and that's because they've got this imbalance in their immune system. They've got lots of one type of white blood cell that can't do its job properly. And also they can experience easy bleeding or bruising because they have fewer platelets. And so this means that these platelets can't clot the blood quick enough in order to stop the bleeding. So what are the main challenges that face scientists that are trying to find cures for childhood leukemia? We're all different. Although I can't see you, I'm sure if I could see all of your faces, I would soon realize that you all look different. We all have different hair color. Even if we have brown hair, it will be a different shade of brown. We have different eye colors. We have different statures, where some of us are tall, some of us are short. So the leukemia is the same. Everybody's leukemia is different. And so that's a big challenge. Also, we need to think about the fact that the leukemia is our own cell at the end of the day. It's not like a bacteria or a virus, something that's foreign that's entered our body. The leukemia has been derived from our own cells. And also the leukemia cells look like the healthy cells. So we can see here, this is a healthy sample, and we see 2 white blood cells here. They don't actually look any different to these white blood cells in a sample from a leukemia patient. There's just lots more of them. So there are many challenges, and these are just a couple that I've highlighted here. However, despite this, we have got really good actually at curing children with leukemia. We've made massive advances over the recent decades. So this graph is a survival graph. And so along the bottom here, we can see the number of years since diagnosis, with the time of diagnosis being at time point 0 here. Up this axis, we have survival. So on the day at diagnosis, everybody here is alive, and so survival is 100%. But over time, what we can see, if these lines drop, it means that the number of patients dying of their leukemia is increasing over time. So in the 1960s and 1970s, we were only able to cure about 1 out of 10 children with leukemia. But as I hope you can appreciate that over time, as we go through the decades and the years, this line has significantly increased. And so what this means is that we have got much better at curing children with leukemia. And so now in the 2020s, we are exceeding 90% survival. So that means we can cure now 9 out of 10 children. So how have we done that? It may be surprising to hear that we haven't actually developed that many new drugs during this time. So we're still using the same drugs that we would have used in the 60s and 70s today. What we've done is that we've, through clinical trials, so by treating children collectively on clinical trials, collecting their data and having access to samples, we've really been able to look at the leukemia. We've been able to look at whether patients respond to certain types of treatment, and we've really been able to make changes and treat patients more effectively during this time. So I'm going to give you a couple of examples of how we've made these improvements. What we have realized is that age is really important and is linked to outcome. So we can have 3 groups here. We can have those that are less than 1 year old at diagnosis, those that are aged 1 to 9, or those that are older than or 10 years or older at the time that they're diagnosed. And what we were able to see by looking at data from clinical trials is that age is associated with outcome. So now we're looking at a graph that considers event-free survival. So again, with time along the bottom here since diagnosis, at time point 0, none of these patients have had an event. So by an event, what we mean is that either they did not respond to treatment and they sadly died of their leukemia, or they responded to treatment initially, but their leukemia came back, so they relapsed. And what you can see is that as the number of events add up, this line kind of comes down. And so those patients that are under 1 year of age actually have a worse outcome. So more children will relapse of their leukemia or will sadly die, compared to those that are aged between 1 to 9 years at diagnosis that actually have a better outcome. While some of these children will relapse and sadly die of their leukemia, far less of them do. So there is an association with outcome. And as soon as we were aware of the fact that age played a role in outcome, we are now able to better or more informatively treat these patients. So if a child is under 1 year of age when they are diagnosed, they will be treated more intensely. We need to treat them more intensely in order to increase their survival, but we wouldn't treat them in the same way that we would treat a child aged 1 to 9 years, because we know that naturally, because of their age, they are going to have a better outcome. So we don't need to treat these patients so intensely. So another indicator of outcome is the number of white blood cells that we can see in the bone marrow at the time that the patient is diagnosed. So here we've got 3 samples from different individuals. So in the top here, I'm trying to make it out like you're looking down a microscope and looking at a sample from some bone marrow. And in this healthy individual, we've got all those different cell types that I told you about at the beginning. We've got lots of red blood cells. We've got these little golden nuggets that are our platelets, and we've got some different white blood cells in here as well. Then we could have 2 samples here from patients with leukemia. And we can see a difference in the number of white blood cells. So some patients can present with a low number of white blood cells, but again, we've still got that one type of white blood cell that is persisting and it's blocked in its development, but there's fewer numbers of them. Or we could have a patient that has lots of this one type of white blood cell. It's really crowding out the bone marrow. And so again, when we've looked at data from clinical trials, we can see that those children with a high white blood cell count, so lots of white blood cells in their bone marrow sample at diagnosis, have a worse outcome. They are more likely to relapse of their leukemia or sadly die, compared to those children who have a lower white blood cell count at diagnosis. So now we've got 2 things to consider. We've got the age of the patient to consider and the number of white cell counts, white blood cells that they have at diagnosis. And so we can start to move the children into better treatment arms of treatment trials to more effectively clear their bone marrow of these leukemia cells. So that's 2. The third, we've actually had to look at the genetic code. So what is the genetic code? I like to think of our genetic code as a recipe book that we would have in our kitchen. So our recipe books have both, they consist of instructions or the method and ingredients. And as long as you read the correct instructions and get the correct ingredients, you should have a meal like this at the bottom, and I would certainly eat that myself. Our genetic code is the same. It's like a book. It's a code from beginning to end, and it has regions that produce instructions, and these are called enhancers and promoters, and the ingredients of our genetic code are called genes. And as long as we have the correct number of instructions and ingredients, and they're in the correct order, so they're on the correct page of this book of our genetic code, then we have a healthy human being. However, if we just go back to the recipe book, if we accidentally pick up the wrong instructions or the wrong ingredients, we may end up with a meal looking like this, and you certainly wouldn't catch me eating that meal. But this is what can also happen, and this is what happens in the genetic code of children with leukemia, is that sometimes we can get the wrong instructions matched with the wrong genes. We can have too many instructions and we can have too many genes. And when that happens, it signals for these cells, so these white blood cells, to start dividing really quickly, but to stop in their development. And so this is when we can see the accumulation of these leukemia cells in the bone marrow. So we've needed to learn a lot about DNA, or our genetic code, over the time. And so quite simply, our DNA, as I'm sure many of you have seen it depicted in the news, as a double helix. So it's two strands wound round each other. But simply, it's just a code of 4 letters. It's 4 letters, A, T, C, and G, all in different orders from beginning to end. What I find miraculous is that this genetic code in every single cell of our body is 6 feet long. So I'm 5'9". If I stretch my arms out, and you can't see from finger to finger, but from the middle finger on my left hand to my right hand is even less than 6 feet. But somehow we managed to fit DNA, or our genetic code, that is that long into every cell of our body. And so then if you look at the back of your hand, you can't even see a single cell. So how on earth does our body get 6 feet of DNA and squash it into a cell that we can't even see? So it has this really cool way of taking our double helix and coiling it up and coiling it up again, and just keep coiling it. It keeps coiling it around itself. And so it condenses and it shrinks this 6 feet of genetic code into a really small space. So the best thing I can think of to describe this is like the cable on a landline phone. If you've ever gone to pick up this phone but brought the whole phone with you, that's because the cable has super coiled itself. And this is what our genetic code does. So when cells need to grow, or when we need to replace cells because they're damaged, we need to take a single cell and it needs to divide into 2. And if we think about our leukemic cells, they're doing this really quickly. We're forcing them to divide really quickly. Only the genetic code could be described as this messy ball of wool. And first thing that we need to do is we need to make an extra copy of our genetic code. So to start off with, we've already got 6 feet of DNA in our cells. Then we need to copy that. So we've now got 12 feet of DNA in a cell. Then we need to make sure that we accurately split that genetic code equally into these 2 cells. And that means we'll have 2 healthy cells come out the other side. But if you imagine pulling this piece of pink wool and then trying to find the end of this bit of blue wool and then pulling it to split it into the 2 cells, you're going to end up with a huge tangled mess. So what you'd logically do is maybe wrap up these different coloured balls of wool into balls, and then you could more easily separate them into the 2 cells, and we wouldn't have this huge mess in the middle. That's what our cells do. So what they do is they package our genetic code into these little structures called chromosomes. And so we could think about maybe having a blue piece of the genetic code that will come into this little blue structure, this blue chromosome, a bit of green genetic code, and so on. So we can condense our genetic code even further into these little structures that enable us then to more easily split this genetic code between 2 cells. And I love to show this photograph. This was actually taken by my PhD supervisor, Professor Christine Harrison, when she was doing her PhD, and she used an electron microscope to take pictures of actual human chromosomes. And these are really cool. So this is the DNA that is all really tightly wound round and made into these beautiful structures that we can see here. So we know that our genetic code can make these structures, but what we didn't know until 1956 was how many little structures our genetic code can be condensed into. And I remember being told this story that I think it was in the early hours in this laboratory that someone could be heard shouting down the corridor, 46, it's 46. And so what they had found is that our genetic code can be condensed into 46 of these little structures called chromosomes. That's an important number to remember. But it wasn't until 1971 when Marina Seabright realized that if you added some dye to these chromosome structures, that you could actually start to see a pattern emerging. So I don't know if you can appreciate, but each of these chromosomes has like a banding pattern. I like to describe it as like a barcode that you would find on something that you would buy in the supermarket. Kind of goes grey, black, grey, black, grey, black. And there's this banding pattern that you can see down these chromosomes. So this was fantastic because now we had a way of seeing if there were any changes in these chromosomes. But what it also enabled us to see is that we actually can start to sort these chromosomes into some sort of order. So we have 2 of each chromosome. So we actually have 22 pairs of chromosomes, and we can order them by size. So chromosome 1 is the longest, and chromosomes 22 are the shortest. And so this is great. So we can start to count them. So 22 pairs is 44. And then if you are a male, you will have 1 copy of the X chromosome and 1 Y. And if you're a female, you'll have 2 copies of the X in every cell in your body. So you should have 46 chromosomes in every cell of your body. And what we can do, since we know that we can now look at these bands on these chromosomes, is that we can identify some errors in the DNA or the genetic code by studying these chromosome bands. So I'm not sure if this next bit will work. So I'm going to ask you a question, and you can probably put your answers in the chat. I'm not sure if I'll be able to see them. I don't want to mess with my screens because I might end up losing you. But Ellie might be able to read out some of them. Don't necessarily need to say names, so people don't need to worry about getting the answer wrong. But let's see if you can be a cytogeneticist for a minute, and these are people that study these chromosome bands. Do you think you could just have a look at this with the knowledge that you just gained from the previous slide? And can you tell me if you can see anything that's wrong here? So remembering the number of chromosomes that we should have, how many of each we should have, and then see if you can identify what's wrong here. I'll just give you a couple of seconds. I don't know if there's anything coming in on the chat, Ellie.
We've not got anything yet. I think I can see what it is, but
okay.
We've got so we've got one person who says 3 of the 21st. Another one that says there's 2 X's and a Y.
Perfect.
Somebody suggested that might be Down syndrome, and there are 4 of the 21 chromosome.
Great. So people are seeing. Yeah, people are seeing exactly the right thing. So what we've actually got here are too many chromosomes. So we should have 2 copies of each. So if we just work our way along, we can see that we've got 2 copies of chromosome 1, 2, and 3. But actually we've got 3 copies of chromosome 4. We've got 3 copies of chromosome 5. We've actually got 3 copies of chromosome 6, and so on. So what this leukemia cell has done, or what we can do by studying these chromosomes, is see that this leukemia cell has actually got too many chromosomes. So that means it's got too many instructions, and it's got too many ingredients when we think about our recipe book. And because of this cell has now got an advantage. It can grow and multiply really quickly. And it can also block the development of this particular B cell. So this type of leukemia is given the name high hyperdiploidy. And if you were to sit and count all of those chromosomes, you'd be able to see that there are 55 of them. And this plus just tells us that each of these chromosomes has an extra copy. So second challenge, this one's a bit harder. Can you tell me what's wrong here? See if you can see what the problem is with these chromosomes.
I don't think yet. I have ideas, but they're probably wrong. So I'm going to wait for somebody else to answer.
This one is harder. And to say that clinical cytogeneticists do this on a daily basis, and they train for years in order to be able to see the changes in these banding patterns. So Ellie, what's your, if no one else has answered, what do you think is going on here?
Well, we've got a few people now. So
okay.
Pairs, and then so some people said they're not matching pairs. Somebody said a possible deletion shorter chromosome. Somebody else said they look short, and not all the pairs are the same and matching. And some of the wrong shape. And I'd noticed that the banding on one was didn't.
Okay. So the reason that they're not identical when we think about the way that we're created is we would have received one of these chromosomes from our mum and one from our dad. So they're never going to look exactly identical. And also it depends how, because we generate these chromosomes by literally firing the cells onto a slide. So it depends how they fall. Some of them can fall at funny angles and they look different. But I'd like to draw your attention to these two chromosomes. So here, this chromosome 4 actually looks a little bit shorter. So some people did say that they saw some smaller chromosomes. And this chromosome 11 looks a lot bigger than the other chromosome 11 sat next to it. What's actually happened here is both of these chromosomes have snapped and they've swapped material. So what we've got now here, rather than this just being all chromosome 4, we've got mainly chromosome 4 with a little bit of 11 stuck on the end. And on this one, we've got mainly chromosome 11, but actually quite a big bit of chromosome 4 stuck on the bottom. So what's really important and what's taken years, even after observing the fact that chromosomes can actually break and swap material, we now need to go into that recipe book, go into our genetic code and understand where these regions of these chromosomes are breaking. And what we find in this particular example is that the genome or the genetic code is actually breaking in 2 genes. So these are like 2 ingredients in our recipe book are being cut in half. So not only are these 2 genes being cut in half, they're actually the half of each of these genes is coming together and creating a new gene. So not only have we cut 2 chromosomes, we've actually formed something new. And what this new gene can go on and do is instruct these cells, as I keep saying, to go on and multiply quickly, or become blocked in their stage of development, or it can even stop them from dying. So it will prevent this cell from dying and it can remain and persist in the bone marrow. So this is another different type of leukemia. So I've only given you an example of 2, and there are many different types of errors in our genetic code that enable us to describe different types of leukemia. And so why is it important that we find these changes? Again, through using clinical trials and samples that we have obtained from patients, we have been able to see that actually some of these changes to the genetic code are linked to a better outcome, and some of them are linked to a worse outcome. So here, the high hyperdiploidy, which was the example that we looked at that had too many chromosomes, when we've looked at clinical trial data, we can actually see that patients with high hyperdiploid actually have a better outcome. So here they track along the top here. So very few of these children will relapse or die of their leukemia. However, when we look at the second example, so KMT2A was one of the genes involved in this swapping of material, we can actually see that type of change to the genetic code is linked to a worse outcome. And that's the case for all of these different changes to the genetic code here that ends up leading to these patients being more likely to relapse of their leukemia or die. So it's really important that we find these changes to the genetic code at diagnosis, because again, like we do with age and like we do with white cell counts, we would change the treatment that we would give to those children. And it really does work. So this graph here shows us data from 2 different trials. The first, the earliest trial, ALL 97, is the dotted line. So there's a red dotted line, a blue dotted line, and a green dotted line. And that was the event-free survival on this trial. And in this trial, we didn't consider the changes to the genetic code at that time because we didn't know that most of them existed. Subsequently, in the next trial, shown by the solid line, red again, blue and green, this is where we actually used that information. So we looked for the changes in the genetic code, and that led to treatment changes. So what we were able to do, and the greatest impact we saw actually were in children that had these genetic changes that were linked to a worse outcome, by intensifying their treatment, because we knew that they weren't going to do as well as patients with different genetic changes. we have managed to increase their event-free survival by 22%. So this was a dramatic improvement. And so now if we put all three together, we can start to see how we have made these improvements in survival over time. And so there are other factors as well. I've only had time today to talk about the three, but that's how we have made these increases in survival. So if I have time, and I might go over by 1 or 2 minutes, Ellie, I hope that's okay. I would just like to tell you a little bit about some of the work that we're doing now that is funded by Ruby's fund. So I'm not going to get to look at these chromosomes because the actual abnormality that is present in some patients that have what we call T-cell acute lymphoblastic leukemia. So this is one type of white blood cell called a T-cell that forms the leukemia in these patients. The particular abnormality here, you can't see it by looking at the bands and looking at these chromosomes. The pieces of material or genetic code that are swapping are actually too small to be seen. But there is, and you're going to have to believe me on this, there is a very small piece of chromosome 14 that is swapping with a very small piece on chromosome 5. So rather than in the previous example, where I told you that the DNA was breaking within 2 genes and we've made a new gene, in this case, what we end up doing is bringing a gene from chromosome 5, we pick it up and we put it on chromosome 14. Only now we've put it next to a really powerful super enhancer. So our enhancers are things that provide instructions. So if we go back to thinking about our recipe book, but this is super. This is a really important, really strong enhancer giving really strong instructions. So we've now put a gene next to this super enhancer, and this gene shouldn't be switched on in a healthy cell. But now it's next to this super enhancer. This super enhancer communicates with this gene and actually switches it on. And the switching on of this gene contributes to the cancer. So we want to understand how these super enhancers manage to switch these genes on. So what we've been able to show is that the way that they do this is that these super enhancers can paint proteins over the gene. And what that does is it physically forces the DNA to open. So rather than being this tight coil here where nothing can get into it, it opens it up. It literally, these proteins force the DNA to open, and this enables the gene to be switched on. So we've taken a further look at these super enhancers, and as I said, these are really big and powerful instructions. And so we thought, how on earth are we going to start to study these because they're so big? But what we have realized more recently is that actually we think there are only actually small regions of these super enhancers that are important for communicating and opening up this gene. And so that's given us slightly smaller regions, more of a slightly easier task. It's never easy, but a slightly easier task in trying to prove that these parts of these super enhancers are important in opening up and switching on this gene. So what we want to do now is stop these parts of the super enhancer from opening up and switching these genes on. And we can actually, whilst the super enhancers paint proteins that activate and open, we can paint proteins that actually shut down pieces of the DNA. So what we're now doing is we're painting proteins over these specific sites in these super enhancers, and we're seeing if we can switch this gene off. So we're seeing if we can shut this DNA back down and switch this gene off. And so the final slide, I'm actually going to show you some really recent laboratory data where we have actually managed to switch the gene off by painting proteins over one of these sites. So the way that we can look at whether a gene is switched on or not is using a technique called Western blotting. And what we can see is when a gene is switched on, we should see a black signal. So we can see black signals here, and this tells us that this gene is switched on. So when we don't see a black signal, what it tells us is that this gene isn't switched on, so it's switched off. So in this particular experiment, what we do is we paint our proteins on this one site of interest in our super enhancer, and then we collect samples 8 hours, 24 hours, and 48 hours after we've done our painting. So just to keep in mind that this painting isn't permanent. So whilst we put this paint on and we hope to shut down our super enhancer, this paint will wash off after a while. So it will stop. What we're trying to do, it isn't permanent. But what we can see when we paint these proteins on this one site in this super enhancer, after 8 hours, we've managed to switch this gene off that is contributing to the cancer in these particular cells. It stays switched off up to 24 hours, but at some point between 24 and 48 hours, our paint in this area is starting to wash off and this gene is starting to be switched back on. So this means our super enhancer is gaining its powers back again, and it's able now to paint those proteins back over the gene and open it back up. So whilst this first experiment, we've only been able to switch the gene off for a small amount of time, as soon as we've identified a small number of sites that can do this, we're going to look to develop experiments where we can switch this gene off permanently. And what we hope we will do is by switching this off permanently, we know that these cells are addicted to this gene. They need this gene to be switched on to stay alive. So we hope that if we can switch this gene off permanently, we can actually end up killing these cancer cells. So that's our ultimate goal. So I would just like to thank you for your attention. It's really hard giving a presentation when you can't see anyone. So I hope that you haven't been thoroughly confused by what I've said. But I would also like to say thank you to patients and their families, and also to fundraisers. Whilst as scientists, we've been able to make these changes and increase the survival together with clinicians for children with leukemia, we can't do our job without patients and families allowing us to use samples or collect clinical data, and we certainly can't do our work without fundraisers that enable us to be able to do these experiments. So the reason we have made this dramatic increase in the survival of children with leukemia is a big collective effort from everybody involved, from scientists, as I say, through everybody involved in this whole process. So I thank you for being part of that and making our jobs possible. And sorry for overrunning by 5 minutes.
No, don't worry about it. Really, really interesting talk. I know I've got a whole load of questions. So we've now reached the Q&A portion, so please do submit your questions, but we're not going to go for mine because they're probably boring. So we have got a question in from someone anonymously. So they've asked, you mentioned high versus low white blood cell count at diagnosis and survival rates. I've often wondered whether this is affected by the time it takes the child to get their diagnosis, i.e. the longer it takes the diagnosis, the higher the count found. We were just over the threshold between regimen A and B and wondered if my son was diagnosed earlier, would he have been on regimen A?
Yeah, so I mean, that's impossible for me to say as a scientist and not somebody who is a clinician, but you are totally right that this white blood cell count is only one snapshot in time. It's the point at which that sample, that bone marrow sample, is taken, and that white blood cell count could be very different even the day before and the day after. So the decision on how that child is treated is taken at the time that sample is removed and the white blood cells are counted. But whether that in your specific case, I'm not able to answer whether that would have moved your child from regime A or B or vice versa.
Okay, so got another question from Kadu. It says, what's the role of microRNA for diagnosis and prognosis of leukemia among children, if you have any further information?
Yeah, so microRNAs are quite understudied. So these are another part of our genome. So maybe they're a different part of our recipe book, but they can also provide instructions, and they're really important in switching genes on and switching genes off. So in relation to kind of the getting into the clinic, we're way behind on looking at microRNAs, and we've really only been able to find them and find where they are since we've been able to sequence the human genome. So I think microRNAs are definitely an area of research that is active, and I think we'll probably be able to find a lot out from it, but being able to translate that to the clinic and make any kind of clinical changes, that's to come.
Okay, so we've got a couple of questions about the painting of the genes. So Somebody's asked if you could explain what you mean by it, and also somebody else wanted to say both at the same time, because I think they're kind of connected. They've asked what side effects you might get, but I wasn't sure if this is something you're just using to explore the genetics, or if it's something you're thinking might be a treatment further down the line. So yeah. Okay,
so for painting the proteins, so our genetic code is the same in every single cell of our body. So if it was only the genetic code that told our cells what to be, we would be made of one cell type, because that code would just tell our cells what to be. So the reason that we can produce all the different types of cells that we have in our body, so heart cells, lung cells, brain cells, breast cells, any of these different cell types, is because we have another layer to our genome. And this another layer is formed by different proteins that combines the genetic code, and different combinations of these proteins. So it's not just one protein. So what we can have in regions of the genome that are active, so maybe a gene that is switched on, we will have proteins that bind to that genetic code that keep it open. And by keeping it open, it allows that gene to be switched on. But if we want a gene to be switched off because, say, we don't want this cell, we want this cell to be a heart cell, so we need to switch on genes that are going to tell our cell to be a heart cell, but we actually need to switch off genes that are going to tell this cell to be a blood cell, because we want it to be a heart cell. So what these proteins do by layering on top of the genetic code is enable the correct genes to be open and active in the correct cell type. But I'm sure you can imagine that if then these proteins start getting painted in the wrong places, like we see when a super enhancer starts to communicate with the wrong gene, we're now opening up genes that really shouldn't be open and switched on. And that's when we start to see the problems that these cells go on to proliferate and multiply too quickly. I hope that makes sense. It's quite a complicated thing to explain.
Yeah, I think it's very clear. So next question, very short one, is leukemia always genetic?
Yes. I mean, there are some patients at the minute where we still can't find what the change is in the genetic code. So whilst we have managed to sequence the human genome, there's still gaps. There's still gaps present in the human genome that we haven't been able to sequence. And there are also limitations to the technologies that we use. And so at the minute, the vast majority of patients that are diagnosed with leukemia will have a genetic change that we can find. We might not be able to see it by just banding the chromosomes. We may have to do lots of other techniques where we go really right down to the code itself, looking at the A's, the T's, the C's, and the G's, and making sure that they're in the correct order. But the vast majority of patients that we see today will have a genetic change that we can see at diagnosis. Why that genetic change exists, there could be many different reasons for why that genetic change has happened in that particular patient at that time. But invariably, we do see changes to the genetic code.
Okay, so next question. I will say we were due to finish at 1:45, but do you have enough time for extra questions?
Yes.
More questions are better. So when looking at outcomes, are you only looking at patients who have been treated with chemotherapy, or do your graphs also include those that have undergone bone marrow transplants?
Yeah, so it would depend upon the trial. So the trial will have different regimes, and dependent upon which regime the child was entered into when they were diagnosed will depend upon what kind of treatment path that they took. So yes, it would depend on each trial and when those particular procedures were introduced, because that's the great thing about clinical trials. What we can do is we can introduce new things, and then we can look to see whether that has really made an impact on the outcome and the survival of these children. And that's really the only way that we can do it, is if we can get children or patients with any cancer really onto clinical trials where we can properly study any changes that we make that we think will improve outcomes. So yes, it will include other parameters as well.
Okay, so Susie would like to know whether the siblings of a child who has leukemia are also at risk. I think there might be a little bit of confusion going on between genetic and hereditary.
Yes.
Questions.
Yeah, so in most leukemias, no, there isn't necessarily. It depends if there is a hereditary predisposition, and in some instances there are, but in most, no. Where we can see leukemia develop in a sibling is in twins. So if twins were monozygotic, if it means that they shared a placenta, so they shared a blood supply, what we can see is if one twin starts the process of developing leukemia, and leukemia is very much a process that develops. So whilst I've shown you some big changes to the genetic code, usually there's lots of little changes that happen and they build over time until the collection of changes mean that these cells can suddenly transform, which means they can suddenly start proliferating really quickly. And so what we can see in some monozygotic twins is that the first change happens in one twin, and then that cell, as it's dividing, some can transfer into the second twin, and then these babies can be born. And then maybe one twin might go on to develop leukemia, and that's because their genetic code has received some more hits, some more changes. And there's the other twin, whilst they've got almost some cells with some changes, those changes aren't enough to trigger the leukemia. So we've studied more in twins where we can actually see where cells can transfer from one baby to the other. But yes, there is a big difference here between having a hereditary predisposition and then just acquiring these changes throughout life.
Okay, so we've got a couple more questions on that sort of vein. So I've got one person who'd like to know if it is genetic, how can people recover? And another saying, if it's genetically driven, is chemotherapy able to reprogram the bone marrow to stop that mutation from reoccurring? So similar questions.
Can you just repeat the first one, because I took a drink and then my mind went blank?
No problem. So the first one was, if it's genetic, why do people recover from it?
Okay, so this is because these genetic changes aren't necessarily there when you're born. So these genetic changes happen over time, and that might be because, you know, of the lives that we live, what we're exposed to. Everybody has changes in their genetic code. It's why we all look different. That's how we've evolved from, you know, apes into humans, because our genetic code has changed and altered over time. So it's really important that our genetic code does change. The problem arises when these changes happen in genes that are really important for keeping those cells healthy. And so if a change happens sporadically in a gene that's supposed to stop these cells from multiplying really quickly, you've then lost that control. So whilst it is genetic, it's not there in every cell of our body and there from the beginning. It develops over time. And what was the second related question, Ellie?
So the second one was, if leukemia is genetically driven, is chemotherapy able to reprogram the bone marrow to stop that mutation from reoccurring?
Yeah, so chemotherapy doesn't reprogram the genome. What it does is it pretty much just tries to kill all of the cells. So we're not doing any genetic reprogramming. We're literally trying to clear the bone marrow of any cell, really. A lot of the chemotherapy or a lot of the drugs that are given are targeting cells that divide really quickly. And we know that the cancer cells divide really quickly. So that's how they can be a bit more specific in targeting the cancer cells. So we actually just literally try to get rid of them all. So the reason that some leukemias come back is because some of those cells are really good at hiding. So they can hide in the bone marrow. They can be protected by other cells in the bone marrow. They almost become, we can describe them as sticky. So they stick to the bone marrow, and they're really hard to treat, those cells, when they're stuck and protected by other cells. So the reason that sometimes leukemias come back is because the drugs haven't managed to clear all of those leukemic blasts, and it only needs to be one that is left behind that will have the ability to become 2, to become 4, to become 8, and start the process again of kind of repopulating the bone marrow.
Okay, so next one from Lewis is, says he was just wondering about age being a factor in determining outcomes. Is there a reason in particular why children 1 to 9 have better prognosis than children above the age of 10?
Good question. No, not really. Sometimes it is linked to the type of genetic abnormality that we see. So especially in those children that are aged under 1, so their age is already impacting their outcome. So they naturally have a worse outcome because they're under 1. But if that wasn't enough, they also go on to get the types of changes to their genetic code that are also linked to a worse outcome. So they get that first swapping of material that I showed you between chromosome 4 and chromosome 11. That's really common in children who are under the age of 1. And so so they've almost got what we would call 2 risk factors there, their age and the type of genetic or alteration that they have to their genetic code. In the children aged 1 to 9, it's again, we can see a real spread of those different genetic changes, but actually we see high hyperdiploidy. So the example I gave you where there were too many chromosomes, that's actually found more predominantly in children aged 4 to 5. So that's probably driving why quite a lot of those aged 1 to 9 do quite well, because they will have changes to the genetic code that are also linked to a better outcome as well. So it's a combination. It's never one thing in isolation.
Okay, so next question is from Lucy. So she'd like to know if an infant is diagnosed, if an infant is diagnosed as an infant, are they more likely to have been born with the bad gene, as her son was 5 months old at diagnosis?
Yeah, so I can't say for every individual case, but what research has shown us is, so when a baby's born, they have the heel prick test, so a blood spot taken from their heel, and we use that for testing for other genetic diseases. But what scientists working on leukemia have been able to do is to look at that blood spot and see if the genetic change that is found at diagnosis was present in that blood spot when that baby was born. And for certain types of genetic changes, we can see them in the blood spot, which means that actually that particular genetic change happened when the baby was growing inside the mother. It doesn't necessarily mean that they had leukemia at the time, because as I said, the development of leukemia is a stepwise process. So that may have been the first step, but there may have been other changes to the genetic code that were required for that cell to then become leukemic. But in some cases, we can see the first hits to the genetic code present in the blood of newborns.
Okay, so next question, if that's okay.
Yeah.
Obviously, I don't think you're in clinic, are you just?
No, I'm just at work because I have young children, and I feared that if I did this from home, they would probably be appearing on this. So I've stayed at work. I don't need to rush off. I'm fine.
Okay. Somebody would like to know how long you should monitor a child who is in remission.
Oh, how long is a piece of string? I don't really know the answer to that question. Most relapses that we see happen early. A lot of them happen in the really, in the worst outcome situation. They happen while the child is being treated. So it kind of means that leukemia isn't responding to the treatment, because we can't even clear the bone marrow of all of those leukemia cells in the first place. So a lot of relapses happen quite early. And we do know that if they happen early, it's much harder to salvage, what we call salvage. So to treat that child for a second time and cure them of their leukemia. If there's a bigger time between diagnosis and relapse, and it does happen, we do see late relapses that happen, but actually in those cases, it's slightly easier to salvage and treat those patients for a second time and hopefully cure them of their leukemia. So there is a difference, but the time at which you stop, that's unknown really, because like I say, it could just be one cell that sits there, and it could sit there dormant and quiet for quite a few years, and then it may get another change and then the whole process can start again.
So in a somewhat linked thing, I'm liking the linked questions today. So Charmaine would like to know, after 5 years post-treatment of survival, do you have the same chance of relapse or getting leukemia that someone who had never had leukemia would have?
Yeah, I mean, I would say that I think you are always at more of an increased risk than a member of the general public that hadn't had leukemia. But I think in the vast majority of cases, having got to 5 years and having seen that out, then obviously the treatment worked, and it worked really well. Again, I don't think we could say that that child would never relapse later on in their life. I think they would always have a more increased risk, but I guess it depends where those genetic hits were in the first place. So I think that's really hard to say. But I guess we have seen relapses later on than 5 years.
Okay, I think we might be on our very last question. So Lewis would like to know, is there a reason why the chromosome translocations have a worse outcome to hyperdiploidy?
So I gave you, so that was one example of how too many chromosomes can actually have a better outcome, but we can actually see leukemic cells that lose chromosomes, and actually that's linked to a worse outcome. So it's not necessarily the translocation or the physical swapping of material. We can actually lose chromosomes, and that leads to a worse outcome. What's driving the worst outcome is what's happening at those locations in the genome where it's breaking. So if it's genes, or if the new gene that is formed is particularly powerful or particularly good at allowing these cells to multiply much quicker, then that is going to always lead to a worse outcome. Or if it enables those cells to evade a drug that we give them, because some cancer cells can be super intelligent that you treat them with one drug and they find a way of evading it. They develop ways of completely ignoring that drug and going on and developing in a slightly different way. So it really depends, and it's not just that in the case where you have too many chromosomes, it's going to be better, and if you swap material, it's going to be worse. You can have swapping of material between chromosomes, and those patients can have a better outcome.
Okay, so I think we have reached the end. So just before I read out my end bit, I'm just going to read out a comment for you, Lisa.
Okay.
So somebody has said, I can't express my gratitude to people like you who are literally superhumans to parents like us by doing this research. Absolutely incredible, and the future looks bright to pushing survival rates further.
Oh, thank you very much. That's a lovely comment to read.
So to everyone else, thank you very much for joining us this evening and for your questions. So the talk has been recorded and will be sending out in the next few days if you'd like to share it or watch again. You will also be receiving an email shortly for your feedback on today's talk. This is so we can make sure our talks are the very best they can be. So please do take a couple of minutes to let us know what you thought. And have a lovely evening, everyone.